correlation between a biomarker and a clinical endpoint of interest, for example, in an epidemiologic study or in the placebo arm of a clinical trial, is not sufficient to support that biomarker can reliably predict clinical benefit and confirm the effectiveness of a drug. Ultimately, the support for use as a surrogate endpoint depends on evidence coming from multiple large long-term trials showing that drug effects on the biomarker can reliably predict drug effects on the clinical endpoint. So focusing just on, say, quantitative MRI as an example, we'd want to see results across multiple trials showing a reliable relationship between effects or lack thereof on a specific MRI-based endpoint and drug effects on, say, long-term development of sustained severe pain or severe function or joint replacement. Unfortunately, at this time, the available evidence supporting the biomarkers that have been discussed today is largely limited to studies that show some degree of correlation between the biomarkers and the long-term clinical endpoints, for example, in epidemiologic studies or in the placebo arms of clinical trials. And therefore, and also considering the complexity of the disease that the speakers described, including Nikolai, I don't think the evidence is currently sufficient to understand the duration or the magnitude of the effects on any of these biomarkers that would be necessary to reliably predict direct benefit to the patient in the long term and to ensure that those benefits will exceed the drug risks. So to address the significant unmet need and to bring safe and effective therapies to OA patients, we really need long-term controlled trials to reliably evaluate the effects on long-term measures of how patients function and feel. And I'm really looking forward to the next session discussion to advance possible approaches related to this topic. And importantly, such trials will not only reliably evaluate the benefit risk of new potential disease-modifying drugs, but they'll also help further develop biomarkers, including opening the door for potential future use of biomarkers as surrogate endpoints as knowledge and evidence grows. And my final comment is I just really want to emphasize the points made in multiple talks that biomarkers can have many other very important uses in drug development other than their use as surrogate endpoints in phase 3 trials. And there are different expectations around the types of evidence that's needed to support different contexts of use. And for certain biomarkers and certain contexts of use, sufficient evidence may already exist. So just to name a few possible alternative roles, there could be value in advancing OA drug development for use as early phase proof of concept studies, for use as diagnostic or prognostic purposes to identify patients to include in trials, for evaluation of safety, for use as baseline covariates for adjustments in statistical analyses to improve feasibility. These are many alternative uses, and I really encourage further consideration and discussion on these. So thanks very much. I'll stop there and looking forward to the rest of the panel discussion.